I'm going to do, I hope this is a forward-looking talk, but I'm going to seize upon examples that have worked, hoping that those examples will provide some conviction in the audience that this process is productive and that we have much to look forward to. So with that in mind, the principle of the KIT receptor tyrosine kinase oncoproteins, of course, has been underscored very nicely this morning. And what I want to highlight is, how did this happen? What were the series of events that caused Hirota et al. To say, gee, it's KIT. It's KIT might be the driver in GIST. And this is a relevant point because we can all look forward to a bevy of new candidate targets coming out of the next generation sequencing, other high-throughput screens that are engaged in sarcoma at the moment and will be extensively so increasingly over the next few years. But how does one identify the gold, the leads that will really pan out amongst many other things that might be okay possibilities, but won't be life-changing for sarcoma patients? So what did Hirota et al. Do? Well, the series of events were that the normal cell counterpart, as was discussed this morning for GIST, was identified as being not the smooth muscle cell, but those interstitial cells of Cajal that drive peristalsis of the GI tract. At the same time, this group in Japan that made the observation in GIST of KIT mutations, they knew that KIT, this receptor protein, was absolutely fundamental for the normal development of interstitial cells of Cajal. If you inhibit KIT in embryonic mice, those ICC cells don't pattern and proliferate in the gut, and you have a sick mouse with a non-functional gut. And they determined that the KIT protein, which was strongly expressed in interstitial cells of Cajal, were equally strongly expressed in GIST, whereas that's true of very few normal lineages in the body. So they say, gee, it drives ICC cells to grow and to survive. Maybe the same is true in GIST. And they sequenced KIT in GIST and found out, of course, that's exactly the case. They identified KIT exon 11 mutations in GIST. So let's just keep in mind that it's often a confluence of perspectives, developmental biology, different approaches to screening, come back to this RNA, DNA, protein. As long as you don't get lost in it, then typically the more perspectives one has to draw upon, the better, that might help you identify the most convincing target. Okay? So we thought this was very convincing, and Marcia Lux, terrific Harvard medical student, came into my lab for 2 months, and she quickly discovered that KIT exon 11 mutations weren't the whole story. There were also exon 9, 13, and 17 mutations that we heard about earlier on. And David Tuvison, also in our group, then took advantage of cell lines that we'd established. And I'll just say parenthetically, when we established the GIST cell lines, we had no particular use for them, but we thought, we hate to see an opportunity go by. Here's a good surgical sample. The cells are growing in early cultures. We karyotype tumors. Let's keep them growing. They might be useful for something. So they were sitting and waiting, a biologically credible model, when this concept developed. And David could then easily take a drug, Imatinib, which had just proved itself for BCR-ABL as an inhibitor, and show that it shut down KIT in these cells and it stopped the cells dead in its track. Not just blocked proliferation, but caused apoptosis. Okay? So one point that I'll say this twice in different ways, but we focused on the heterogeneity of sarcoma histotypes, at least 70 different types. But within those subtypes, if we break them down into molecular subtypes, we end up with things that may be kind of homogeneous. And if you have part of the explanation, a target in one portion of one of these relatively homogeneous final subtypes, but it's not there in the other, that's often a starting point to discover what is the target for the remainder. Okay, let me make that more clear. In the case of GIST, we knew quickly that there was a small percentage of these tumors that lacked the KIT receptor tyrosine kinase mutations. And because the rest looked kind of like the ones with KIT mutations, they weren't wildly different histologically and clinically, we thought, well, maybe they have a similar sort of thing. So what we did is we took some of those GISTs, we had nice frozen materials, we purified out of it everything that was a phosphorylated, a tyrosine phosphorylated protein. Phosphorylation is often the hallmark of activation. Then we took that protein fraction and we purified from that everything that was a tyrosine kinase. KIT is a member of the receptor tyrosine kinase family. We used mass spec, and lo and behold, what we found was lots of PDGF receptor alpha, platelet-derived growth factor receptor alpha. So Mike Heinrich, Chris Corliss, our collaborators, then sequenced PDGFRA in those cases and quickly found that there were mutations just like those in KIT. So that leads us up to the slide that Drs. Berman and Rout both showed earlier today with these various different mutations. Okay? So another example. This is example number 2 of taking a subset molecular subset where you thought they'd all be the same, but they don't turn out to all be the same, as a basis for discovery of a different sort of target. Here's a not unusual dramatic clinical response of a KIT exon 11 mutant GIST to imatinib over a period of 6 months. And here's the survival curve that is the correlate for that, going out to about 2 years. This was the first phase 2 study of imatinib in GIST, and you can see with the exon 11 group, the survival is pretty spectacular. Mind you, these early patients had a huge burden of inoperable disease. They're quite different from the patients who were often first treated on imatinib today. And, the survival's great, but there are a few patients who progressed early on and who actually, this is overall survival, died of their disease. Okay? So we've been fascinated in that group. They have a drug-sensitive target, and we think that KIT is not just an important mutation, but the initiating mutation in these tumors. Gee, why don't they respond to the drug? So here's a patient. We're just working up this patient in the past month, but I'll show as a case in point. This was a patient with a 24-centimeter GIST, showed a mixed response to imatinib, where some of the GIST seemed to respond and some of the disease was clearly progressing. So it was taken out at the 6-month time point. And we have parts 403 and 404, which are different aspects of the progressing aspect of the GIST. In red, we have the primary KIT exon 11 mutation, which we know is sensitive to imatinib on its own. And what we have in 403, in addition to the KIT exon 11 mutation, is the first KRAS mutation that we had found in GIST. And then in this part, 404, we have not one KIT mutation, but 2. And this KIT T670I is a so-called gatekeeper mutation. For those of you who know the CML story, where there's a very consistent imatinib resistance mutation in BCR-ABL, this is the exact correlate to that amino acid change in the KIT receptor tyrosine kinase, the same thing we see in ABL. Okay? And because Dr. Raut is such a pillar of our laboratory, ensuring that the correlative science studies are maximized, we usually have good frozen material to provide a correlate for these observations. So what did that show? Here's the Western blot. In 403, which had the KRAS mutation, the target of the drug, KIT, so this is phospho-KIT, activated KIT, it's fully shut down. But the cells don't care because KIT's irrelevant. You're inhibiting the target at the cell surface, but KRAS, which is just downstream, is on, and it's firing off growth and survival pathways to keep the cells running. They no longer care much about drug. 404, which had the new KIT mutation, the gatekeeper, you can see that KIT, phospho-KIT is strongly present, so KIT is activated in the presence of drug because this mutation changes the structure of KIT so the drug can no longer shut it down. And then, by the way, we see with the KRAS mutation, you're all familiar with the catechism, or some of you are, RAS, RAF, MEK, MAP kinase, which isn't quite as linear a pathway as it sounds, but it's not happenstance that MAP kinase is strongly phosphorylated in the presence of this KRAS mutation. Okay? So I'll skip over these 2 slides because Bob Mackey discussed them beautifully. I just want to make the point that good old-fashioned techniques like classical karyotyping continue to show new chromosome rearrangements, which when one finds out what genes are targeted by those rearrangements can lead to new drug concepts. So in the case of dermatofibrosarcoma protuberans, where you often see a ring chromosome, and the key thing is that juxtaposes collagen 1 alpha 1, gene very actively transcribed in a myofibroblast kind of tumor, with PDGF beta, the ligand for platelet-derived growth factor receptor beta. Then you get autocrine paracrine activation of PDGF receptor beta. That's an imatinib target, not as quite as good a target as those mutant forms of KIT, but a good enough target that one sees clinical responses in DFSP. And then a more recently reported one, this is a inflammatory myofibroblastic tumor, typical of the ones that one sees in kids and younger adults that have ALK expression. Again, how was this discovered? It was actually another student in my group who took a few chromosome translocations in IMT and showed that they all hit ALK, resulted in ALK fusion genes. So brown immunostaining for ALK strongly expressed. These spindly cells are the neoplastic cells in the IMT, just like the diffuse giant cell tumors that Bob Mackey discussed earlier today. Around those cells you have a rich inflammatory infiltrate, which is reactive, which doesn't express ALK, but it's been reported these respond very nicely to crizotinib and other ALK inhibitors. Okay. So now turning to high-throughput screens, and I'm not going to show examples of each of these, but just to buttress the point that I made earlier today, whether one is screening at the expression level, the transcript level, so it might be gene expression arrays, or we're incredibly enthusiastic at this point about transcriptome sequencing, which still costs more, but it gives you expression in a very nuanced way, plus the mutations. Or genome approaches, medium resolution approaches where you scan scattered points in the genome, CGH or SNPs, or high resolution like exome sequencing or whole genome sequencing, proteomic approaches, functional approaches. I have RNAi. What I don't have here are chemical screens, where you just go for the jugular. You say, we've got a good cell line, biologically credible, let's screen a drug library and see what shuts down the cells. Okay? But, again, to make the point, if you can do it, then more than one of these is better for obvious reasons. Okay? So now I'll turn to a few examples of high-throughput screening. This is a slide that Jordi Baratina gave me, so Jordi's is the only name on the slide, but I want to emphasize this was a collaboration, as we see down below, between Memorial Sloan Kettering, Sam Singer, Mark Ladani, Bob Mackey, and others, and Matt Meyerson's group at Dana-Farber, where very ambitiously, and the technologies move along with incredible pace, but when this was initiated, I think it was 3 or 4 years ago, it was almost mind-boggling. This was an interrogation of 48 soft tissue sarcomas. You see the types listed up above. And the analyses were at the sequencing level, all the tyrosine kinase genes. We know those are oncogenes. We know many of them are druggable. Let's find out if some of the sarcomas have those. And then a number of other cancer and sarcoma-related genes, all the microRNAs. And the following slide just summarizes some of the highlights. So one that's very interesting, in the myxoid round cell liposarcoma group, 18% have PIK3CA mutations, so clearly a very druggable target. And then there's other examples, P53 mutations that we didn't know about in pleomorphic liposarcoma, neurofibromatosis type 1, in myxofibrous sarcoma and pleomorphic liposarcoma. Not quite as straightforwardly druggable, but certainly with opportunities. And another study that was done, this is again the medium resolution genomic screen, whereby SNPs, one is asking, what are the regions that are highly amplified, way too much of that in the genome, and what are the regions with localized deletions? So here in the dedifferentiated liposarcoma group is the well-known amplification that grabs MDM2 and CDK4 on chromosome 12. MDM2 has been a bit of a frustrating target to drug in the early going, but we have high hopes for that. And there are a number of other gains and losses, if you look up the paper in Nature Genetics, in these various types of sarcomas that look very promising. Okay? Here's another high-throughput study. This one was done at the Dana-Farber, and so we included a group of 96 sarcomas, a different approach, a mass spec approach called Sequenome, which nonetheless interrogates particular nucleotide mutations. So we're not interrogating the genome, we're saying we know that there are certain mutations that are highly repetitive, like the KRAS mutations, the BRAF V600. Let's screen for those in a whole lot of cancers. So there were thousands of cancers that were included in the screen, and as I said, 96 sarcomas. So here's the summary slide. Pink represents mutations that were found in sarcomas. You can see that all the KIT mutations were found in sarcomas. Not a surprise. We put some GISTs into the series, and none of their other thousands of tumors had the same sort of KIT mutations. But I think very cool, we also see some HRAS mutations, which included some rhabdomyosarcomas, some BRAF, one of those being a rhabdomyosarcoma as well. And why is that cool? Because there's a lot of evidence that shows that activation of RAF-related events is a fundamental part of many rhabdos, especially embryonal rhabdomyosarcomas. And Christina Antonescu and colleagues, with collaboration from Memorial Sloan Kettering, with collaboration from our group, has shown that you get a kind of phenocopy of that in GISTs when they're escaping the selective pressure of Imatinib. Rare GISTs to be sure, but this happens occasionally. So the starting point for this patient was an epithelioid type GIST with a PDGF receptor alpha mutation, expressed KIT and PDGF receptor alpha. At progression, 20 months later, the thing is now very rhabdomyoblastic, and we've lost KIT and PDGF receptor alpha expression, but we have this diffuse, strong Desmond stain, which is absolutely un-GIST-like. This is a myogenic marker. What do we have now? A BRAF mutation. The PDGF receptor alpha mutation is still there as well. It just doesn't matter anymore. It's kind of like when we added a KRAS mutation and took the load off of KIT. Okay, so for the last part of the talk, I'm going to bring some of these techniques together and give you a sense for certainly our enthusiasm for the next generation approaches and how that can make quick work of some challenges that until a few years ago would have been a kind of stop you in your tracks situation. And I'm going to turn to, we haven't reported this yet, but a novel gene fusion which we think is therapeutically relevant in high-grade endometrial stromal sarcomas. The backdrop for this is some work that Jeff Sklar, then in our department, he's now moved to Yale, led with my group about 10 years ago, showing that many of the low-grade classic forms of endometrial stromal sarcoma have a chromosome 7p translocation, and that results in rearrangement of a transcriptional regulator called JAZF1. Okay? That's a straightforward group of endometrial stromal sarcoma. A very contentious group is what we're going to call high-grade endometrial stromal sarcoma, and where the cognoscenti, certainly the GYN pathologists in Boston, argue substantially, are these endometrial at all? Is the evidence really convincing? Or should we call them high-grade uterine sarcoma because it's not convincing enough? We think they're high-grade endometrial stromal sarcoma. What I'm going to show, coming back to a point that Dr. Brennan made earlier, is that establishing a molecular event which is inextricably linked with a subset of tumors, even one that you've been arguing, is it this or that, gives a presence to the entity, certainly makes it something that we can track and we can work with in a much more straightforward manner. Okay, so I'm stuck. Oh, I'll go back. Okay. So we had karyotyped 10 of these, we'll call them high-grade endometrial. They're high-grade uterine sarcomas for sure. They didn't have the 7p rearrangement. What each of the 6 of the 10 had was a balanced translocation between chromosomes 10 and 17. That means both parts of the translocation are there. We got our first surprise when we used standard approaches to define what were the breakpoints of these translocations. The 17 was easy. It's the first oncogene in the so-called 1433 protein family. This is a gene called YWHAE. And I know gene names are boring, but biologically, this is exciting because it's a master switch regulating signal transduction, cell invasiveness, cell growth, and an area of biology there where there's been a huge amount of studies, so it gives us loads to run with, but there's never been a 1433 honest-to-God oncogene where you can point to a mutation and say, no question about it, it's a driver in the tumor. So it gives us a great model to study how these things cause cancer, okay? Now, what was the surprise? When we looked at chromosome 10, we had not one breakpoint, but 2 translocation breakpoints. Not the same case, but as you looked at different cases, the breakpoint could either be here or it could be here, and those are big regions apart. That's like a breakpoint in one state and another breakpoint in another state. There are 500 genes in between. So this was a little bit of a mystery, right? Most breakpoints, it's one gene that you're targeting, okay? The next bad surprise was when we tried to get in close to the breakpoints and map the thing, and we found that we were stumbling around sort of figuratively in the fog. We could not get a landmark. And the reason for this, I'm going to confuse you a bit, but this blue line is not from one of the ESS. This comes from somebody's normal DNA where we've done whole genome sequencing. And these little bars, which we're seeing at low resolution, but down here they're at higher resolution because I've blown the figure up, these are the number of times that each DNA sequence was encountered randomly in doing the whole genome sequencing. And it was mapped by the software to say, it's there, it's on that part of chromosome 10. So, the whole genome should be at about the same height because you're sequencing the genome at 40x coverage. But notice when we get there, there's a big gap. And when we get there, there's a big gap. Where were our 2 breakpoints? There and there, okay? So, to make it simple, nothing is mappable. You can make a DNA probe, use it for fish, you can do race, for the cloning geeks like myself in the audience, but it doesn't work. The techniques don't work, okay? So we went to David Huntsman's group at Vancouver, one of the masters of transcriptome sequencing, whole transcriptome sequencing recently, and they have software that lets you just drill down on single nucleotide differences to say, it's this gene, it's not that gene. What they were then able to do, so this is the high-resolution view of that region right there, is show that this gene, FAM22A, was the fusion gene with the YWHAE. And in this region, there's a almost identical gene, 99% identical FAM22B, which is the other fusion possibility. At the translated amino acid level, it really doesn't matter whether you fuse with FAM22A here or FAM22B there, because the fusion proteins are so identical that functionally we think they're equivalent, okay? And here's RT-PCR, I won't belabor that, from three different cases showing that whether it's FAM22A or FAM22B, you get the same RT-PCR products. You can do a universal PCR, one primer will amplify both of the possibilities. Okay? So what's the story with the component parts for this novel fusion oncogene? Well, YWHAE I introduced before. That's the cytoplasmic protein. It binds to phosphoserine and phosphothreonine motifs. FAM22A is, it's a gene that's expressed in very few normal cells in adult life. It would have been a mystery to us, but we got lucky because the only closely related gene in the genome is called NUT. And NUT was discovered by another postdoc in my lab as the target of another translocation. It happens to be in another rare entity, midline lethal carcinoma, or NUT midline carcinoma, diagnosed in kids and young adults, amazingly aggressive, disseminates in a matter of months. It's a killer of a carcinoma. So we know something about NUT because Chris has continued to work strictly on NUT, and that gives us a sense of wherewithal for this FAM22A. Okay? The odd thing is YWHAE is in the cytoplasm. FAM22A, based on certain structural properties we predict, is in the nucleus. So what does that mean for the fusion oncoprotein? Okay? Well, we confirmed that the fusion oncoprotein here is YWHAE, and this is a cytoplasmic fraction. This is a nuclear fraction. It's almost all in the cytoplasm, whereas the YWHAE FAM22A, that's our fusion oncoprotein. See the band there? It's in the nucleus, not in the cytoplasm, and immunohistochemistry also tells us it's in the nucleus. So it's no question that it's taking YWHAE functions and dragging them into the nucleus. We then asked, what were the interacting proteins? And I won't go through all the steps of the isolation and the mass spec here, but just to say that they included heat shock protein members, which some of you know are critical in ensuring the localization, function, folding, proper management of many oncoproteins, and intriguingly also included RAF1 and BRAF. Okay? Based on that, we've treated our various models with HSP90 inhibitors, and this is 17A-AG, an ansamycin HSP90 inhibitor, STA9090, a new generation synthetic HSP90 inhibitor from the company Synta, and viability. They both have striking effects on the viability of these models. So we think that's a lead therapeutically. The part that we're the most excited about is that there's no question but that this YWHAE FAM22 is dragging BRAF and RAF1 into the nucleus, co-opting normal RAF functions in a way that's very tantalizing. We think that will be therapeutically relevant as well. Okay? The reason that we need therapies, so here's the low-grade classic ESS that I mentioned a while back. They're typically ER and PR positive. They're responsive to hormonal manipulations, whereas these high-grade uterine sarcomas are virtually always ER, PR negative. They don't respond to hormonal manipulations. They don't respond to much of anything. So we do need therapeutic leads. As a biomarker, this seems to have remarkable specificity. You know, always take this with a grain of salt. This was done in my lab. I think it was done very well, but one tends to say a marker has 100% specificity, and then other groups come in a year or two later and pull the rug out from under you. Say, well, not really. Maybe it's 90, 80, 40% specificity. We think this one will stick. So what Cheng Han showed was that in the classic endometrial stromal sarcoma, 0 out of 40 cases had this fusion, and in a total group of 829 mesenchymal tumor, many of them uterine sarcomas, but others extrauterine, then 0 out of 829 have this fusion. We did find the YWHAE in 2 leiomyosarcomas, but not involving a fusion with FAM22. And gene expression profiling, here a nice study. Matt Van der Rijn was mentioned earlier today, and he's one of the gurus of getting gene expression profiles from formalin-fixed paraffin-embedded materials. Here he's used an approach called 3-seq, where you sequence the 3' ends of degraded RNAs from formalin-fixed paraffin materials and done a beautiful gene expression profiling showing that these higher-grade ESS cluster separately from classic ESS and from high-grade uterine leiomyosarcoma. So in summary, this novel fusion, YWHAE FAM22A, promotes ESS proliferation, localizes to the nucleus, interacts with HSP90, BRAF, and RAF1. HSP90 inhibitors and preclinical validations demonstrate efficacy, and we think recognition of this genetic subtype of ESS is important prognostically and therapeutically. I don't have a summary slide for the overall talk, but I want to make the point, I suppose, one more time that we have various ways of screening for new targets, some of them intermediate throughput, some of them extremely high throughput, and having more than one perspective, particularly if it's marrying a genomic kind of survey, which technically is quite straightforward, with the functional surveys, which I would say conceptually they're straightforward, but to do them right is a little bit trickier. But having those two levels of perspective of engagement, the genomic mechanism and then something that's more cell biology related that gives you a better sense of function and druggability, then that's a great approach to take. And with the number of these surveys that I know are being applied in sarcoma, we're going to have many opportunities, much to contend with in the next few years. It's going to be interesting to see which groups have the critical mass, the number of studies, and the sense of opportunity to drive those home the most effectively. But the data flow is going to be startling. Okay? So I'd just like to acknowledge the clinical team, Dr. Rout, Dr. Dimitri, Dr. Fletcher, the other Fletcher, Chris Fletcher, and many others. The lab members who played critical roles were Dr. Wenbin Wu, postdoc, Mark Maeda, Adrian Marino Enrique, and unfortunately I don't have a picture here of Cheng-Han Lee, who did most of the cloning work for the YFAM. He's a terrific young surgical pathologist, sarcoma guru, who did a great postdoc. He's now back in Vancouver building his own sarcoma research group. So thank you to the organizers and to the supporters of this event, which I think is such a terrific opportunity.